Even if excluding origin of life from the scope of Darwinism has a political/historical dimension to it, there still remains the question: can both origin of life and the evolution of life be unified under a single theoretical umbrella? It seems they should. Both are phenomena of life, after all. Why shouldn't they? That said, let's frame the question with a metaphor. Imagine you have a racetrack. There's a starting line which demarcates the prebiotic world from the living world, and lined up in the lanes are various theoretical scenarios for the origin of life. There's Darwin's warm little pond idea. There may be a series of so-called bottom-up scenarios. We'll go through some of those momentarily. Or there may be some other theory. So, which scenario will be the one that drags the prebiotic world over the starting line and into organized life? Darwin's warm little pond metaphor has been a very rich field for cultivating ingenious scenarios for the origin of life. If you want more detail, check out my online course in Physics of Life, Thermodynamics. But for now, let's just take a quick tour. The first of the modern warm little pond scenarios was the emergence of chemical precursors to life, things like amino acids, sugars, and lipids, so-called abiogenesis, prebiotic biochemistry, essentially. By the 1920s, chemists had come to know quite a bit more about these topics than Darwin and his contemporaries did. Geologists also had begun to glimpse the physical nature of the prebiotic world, the composition of the atmosphere, likely meteorological conditions, temperatures, those sorts of things. Around this time, Alexander Oparin, a Russian biochemist, and a British biologist, J.B.S. Haldane, independently proposed plausible models for abiogenesis. We've already met J.B.S. Haldane as one of the founders of the genetic theory of natural selection. He was quite the interesting character. Their scheme for abiogenesis has come to be known as the Oparin-Haldane hypothesis. In a sentence, abiogenesis would come about when energy, in the form of lightning or ultraviolet radiation, say, powered the synthesis of amino acids and sugars from ammonia, water, and carbon dioxide in the atmosphere. These would then accumulate in the ocean. As was Haldane's habit, he coined a clever moniker to describe this: the primordial soup. Primordial soup remained a largely speculative idea until the 1950s, when Stanley Miller and Harold Urey demonstrated that primordial soup could be produced on the laboratory benchtop. They crafted an ingenious experimental apparatus that supposedly mimicked the conditions of the prebiotic Earth that had been proposed by Oparin and Haldane. And sure enough, their apparatus yielded amino acids and sugars. Abiogenesis was quite easy, as it turned out. But did Miller and Urey actually cook up primordial soup? Maybe is one answer to that question. Maybe not is probably the better answer. Among other things, the goodies, the amino acids and sugars, were produced in very small quantities, and they were mixed in with a lot of other stuff. So Miller's and Urey's benchtop concoctions could not by themselves explain much about the origin of life. Soup, after all, is just soup. It just sits there. This conundrum was the starting point for the next big trend in origin of life research. If dilute primordial soup could somehow be encapsulated, this might concentrate it inside some kind of a protocell. Alexander Oparin kicked off the ball here, proposing what he called his coacervate hypothesis. Oparin proposed that when a dilute mixture of simple lipids and perhaps some simple peptides and sugars could be shaken together somehow, perhaps in breaking waves on a primordial seashore, these could form coacervates, self-organized little spheres of lipid enclosing the peptides and sugars. These would not be cells, of course, but maybe they could set the stage for the emergence of cells, or so Oparin thought. Another encapsulation idea came from the biochemist Sidney W. Fox. His small packages took the form of what he called proteinoid microspheres. Fox's proteinoid microspheres began with prebiotic amino acids, which Miller and Urey had found in their cooked-up primordial soups. Under extreme heat, these prebiotic amino acids would polymerize into a structure known as a proteinoid, and these would then form into tiny proteinoid microspheres. Like a coacervate, the proteinoid microsphere could enclose other prebiotic chemicals in an internal environment, and there they might engage in a limited set of chemical reactions. If different microspheres could catalyze different sets of chemical reactions, these could then associate into primitive metabolic networks. These essentially biochemical approaches to the origin of life eventually bumped up against an irreconcilable conundrum, though. If life is characterized by a complex metabolism, what is it that sustains that complex metabolism through time? For there to be evolution, this complex chemistry had to persist through time. Hereditary memory is one way persistence of complex metabolic chemistry can be ensured. So, how then did hereditary memory come about? The conundrum here is that hereditary memory is also a product of complex metabolism. DNA replication, for example, itself involves a complex and highly specified metabolic network. So, which came first? Was metabolism first out of the gate, or was it hereditary memory? This, of course, is a classic chicken and egg problem. Which came first? Each depends upon the other, and it's impossible to identify either as cause or effect. So the next phase of origin of life research focused on ways that both metabolism and hereditary memory could arise simultaneously, a kind of hybrid metabolism/memory sort of approach. The first such hybrid model brought up to the starting line was RNA World, the brainchild of two scientists, Walter Gilbert and Alexander Rich. According to Gilbert and Rich, the first nucleic acid to be cooked up from primordial soup was RNA. RNA had a number of interesting properties that could meld hereditary memory and complex metabolism. It contains a nucleotide sequence code, for example, and this can help guide the synthesis of simple peptides. RNA is also capable of limited self-replication, something which DNA needs an entire biochemical infrastructure to accomplish. And RNA can act as an enzyme, a so-called ribozyme, that can catalyze the rapid conversion of reactants to products, something that proteins mostly do in present life. In this way, Gilbert and Rich proposed that RNA could serve as a bridge to the DNA world of current life. In RNA world, heritable memory, scratchpad memory, and function are embodied in one type of molecule, RNA. As life made the transition into DNA world, only then were these attributes parceled between DNA, RNA, and protein as they presently are. A closely related but ingenious hybrid model for the origin of life comes from Alexander Graham Cairns-Smith, a Scottish chemist whose intriguing model suggested the first living organisms actually were based upon silicon rather than carbon. The stuff of science fiction, that, but actually quite plausible. Silicon crystals, which form clay minerals, have a number of intriguing properties that make them candidates for a kind of silicon-based proto-life. A silicon crystal, for example, can engage in catalysis, that is, specifically directed chemical conversions of reactants to products. There's no mystery about this. We use clay catalysts all the time, the catalytic converter in your automobile being a common example. Silicon crystals, being crystals, can engage in a form of replication. The crystal has a form that serves as a template for its own growth, a kind of hereditary memory. When a silicon crystal grows, an impurity, known as a dopant, can sometimes be incorporated into the crystalline matrix, altering its structure, and this new form can itself replicate, a kind of mutation. These new crystalline structures can also engage in catalysis, but of new types of chemical reactions. Things get very interesting when organic molecules, like those primitive amino acids and sugars in primordial soup, can be dopants. And this can form an entirely new type of crystal, a hybrid silicon-carbon crystal, which can itself replicate. If this new type of crystal can harness energy, such as sunlight, it can then catalyze the conversion of simple reactants into complex products. Cairns-Smith argued that these inherent properties of clay crystals could have formed the foundations for the emergence of complex carbon-based life, and for the simultaneous emergence of carbon-based metabolism and heredity. It didn't happen all at once. The march to life involved a sequential buildup of complexity sufficient to drag proto-life up to the starting line, so to speak. Starting from clay crystals, which could have formed the simple foundations of merged heredity and metabolism, a foundation of hybrid silicon-carbon proto-life forms could gradually be laid down. As this foundation built up, some of the clay crystal proto-life forms would have become unnecessary and could fall away. Think of how an arch can be built. You first build a scaffold upon which you begin to construct the arch, and once the keystone is in place, the scaffold can go away and the arch will stand. Merged metabolism and memory would have then become more complex as hybrid silicon-carbon organisms emerged. Again, the less complex scaffolding could have eventually fallen away, again, like building an arch. And eventually, the carbon-based part could have become sufficiently complex to exist independently of the hybrid silicon-carbon foundation, brought to this level by the arch of complexity built upon the previously established proto-life. Presumably, hereditary memory and metabolism were still merged at this point, and this could have been the starting point for something like RNA world. The point is that RNA world would never have gotten off the ground, so to speak, were it not for the foundation of complexity that had been previously built up by this weird silicon-carbon proto-life. From there, the newly independent carbon-based life could see the differentiation of hereditary and metabolism into DNA, RNA, and protein, the start of DNA world, and the genetic takeover of prior intermediate stages of life. Phew, that was a lot to take in, and I only scratched the surface. That said, I want to make 2 points about going forward toward a coherent model for the origin of life and for the evolution of life. The first is that there has been a great deal of truly ingenious thought given to the question of the origin of life. Whether we're talking about primordial soup, encapsulation, as in the case of coacervates or proteinoid microspheres, to hybrid gene metabolism ideas like RNA world or Cairns-Smith clay organisms, I hope you have come away convinced that a great deal of brilliant work has been devoted to this question. Nobel Prizes have been given to several of the scientists I mentioned. My personal favorite is Cairns-Smith's ideas about clay organisms, an idea, as it said, that's so crazy it just might work. But my second point is this. For all the genius power, none of these brilliant ideas has been sufficient to drag any of them over the starting line into the realm of life. Some have come very close. My bet is that Cairns-Smith's ideas of clay organisms and genetic takeover come the closest. But in the end, all have been prone to a degree of miraculous thinking at the end to help them make the transition into something truly living. No one has yet been able to create life in a test tube, in other words. I'll have some comments on this at the end of this video. But for now, we have to ask, why not? Why have all these ingenious theories not delivered the big prize? One clue is the one thing they all hold in common. They are all inspired in one way or another by the warm little pond idea. That is, they have attempted to explain the origin of life as emerging spontaneously from the molecular to the complex organism. That is, chemistry becoming life, in the words of one prominent researcher in this field. Perhaps then, it's this common feature, from the bottom up, so to speak, that is their common shortcoming. So maybe try a different perspective. And what might that different perspective be? Let's begin by reminding ourselves of what life is. Organized life is not so much defined by its chemistry, but by its thermodynamics, something we've covered in a previous episode. Specifically, life is a specified dynamic disequilibrium where free energy does work on disorganized matter to impose a specified organization and orderliness on it. This is an unstable state, and unstable states always degrade to equilibrium, that is to say, to revert to its disorganized state. For an organism to persist in time, work has to be done continually, cycling matter between its disorganized and organized states. Okay, back to the origin of life. At the molecular scale that prevails in warm little pond thinking, the degradation rate is very fast, explosively fast, in fact. And this means that work must be done at a very intense rate for putative, organized predecessors to life to persist. And that is the problem with the warm little pond. Whether we're talking about primordial soup, encapsulated coacervates or proteinoid microspheres, or hybrid genetic metabolic systems like RNA world or clay organisms, dragging these systems up to life's starting line is going to be thermodynamically very dicey. Even if we imagine some kind of complex network of chemical reactions could come about, it would very quickly be blasted to pieces, so to speak. It could not persist, in other words, and persisting is what living things do. At larger scales, however, it's a different story. Here's a short video clip from a nifty little app called Earth. It takes weather data worldwide and reconstructs global patterns of winds and ocean circulation. This clip is for winds, and the thing I hope you notice here is just how orderly these large-scale flows are. And this underscores a very important point about scale and thermodynamics. At large scales, orderliness emerges more easily than it can at small scales. There's nothing magical about this, it's just how thermodynamic systems at large scale behave. It's a well-established principle, in fact, embodied in something called the fourth law of thermodynamics. The orderly patterns of oceanic and atmospheric circulation you see here are the crest of a standing wave of order production, an idea that we also developed in a previous episode. Free energy in does work to impose orderliness on disorderly matter. This orderly state is unstable and so degrades spontaneously back to disorder, converting the free energy into heat. And the standing wave can persist only as long as energy is continually fed in. This thermodynamic attribute of life has inspired another approach to the origin of life proposed by the Israeli biochemist Adi Pross. A key element of Pross's idea is that life originated by piggybacking onto a persistent thermodynamic regime of orderliness. Pross calls this dynamic kinetic stability, or DKS, which is essentially equivalent to the concept of specified dynamic disequilibrium that characterizes life. Life will originate, according to Pross, by being able to bridge the gap between the chemical and thermodynamic state of dynamic kinetic stability to the specified dynamic disequilibrium of life. And Pross himself expresses this with a colorful aphorism, how chemistry became life. Pross's approach is a significant step up from the miraculous thinking that inevitably intrudes into most bottom-up approaches to the origin of life. But we still have to ask the question, does dynamic kinetic stability get us over the starting line? Let's recast the question in terms of our standing wave metaphor for life. In a purely physical world, there's a frothy crest of orderliness at the top. Pross's claim is that life will begin here, signified by that developing red fringe at the crest. As we did previously, let's take a closer top-down look at that frothy crest where life sits. Again, the problem is how to get dynamic kinetic stability over the starting line to a specified dynamic disequilibrium. Let's fade life's frothy crest away for the moment, and let's imagine some prebiotic mechanism operating near the crest of persistent orderliness. This could be a system of dynamic kinetic stability, or it could be any of the mechanisms that we have discussed so far. The key point I wish to make here is that the transition to life will be easier if it happens at large scale, where the crest is a natural outcome of the fourth law, like those orderly patterns of atmospheric circulation. It will not be possible at small scale, however, where the orderliness is very transient. This pre-biological system can be imagined to bump up against the starting line to make the transition into life, but it actually can't quite get over it. So the question is, what will drag it over? We need three elements to drag the system over the starting line into life. First, the proto-biological system has to have a capability to take in information about the environmental state it sits in. It should also have the capability of taking in information about the system's internal state. Second, there has to be contained within the system mechanisms that can tap into energy and use it to do work to restructure the external state in a way that manipulates a flow of matter that can sustain the disequilibrium state, to enable it to persist. In a word, the extended organism principle at work. Couple the state sensor to the work engine and you have the elements of a cognitive system, which can expand and seek sources of energy and matter from the lower entropy that sits higher on the crest, establishing life at the low entropy crest. And here we have a truly radical hypothesis for the origin of life. Life may have a foundation in chemistry and physics, but what really drags it over the starting line is the origin of cognition. Now, I'm not going to claim that this has solved the problem of the origin of life. Far from it. All I am suggesting is another way to frame the question. Nothing else has worked, so what have we got to lose? I am claiming, however, that this is the unifying framework for both origin of life and evolution of life. Both are phenomena of cognition, life's unique attributes. Oh, a skeptic might say, I've got you now. This whole argument falls apart once someone synthesizes life in a test tube, so to speak. That would prove that life can come from chemistry in a warm little pond, right? Well, not so fast. What such a feat would prove is that given enough intelligence, foresight, wisdom, organization, money, and resources, scientists could pull off something that supposedly happened by itself and unassisted some 3 to 4 billion years ago. So tell me, how is this different from creationism? Substitute the hand of the creator God with the hand of the scientist God, and there really is no difference.